Um, thank you for coming for what might be Ifaz's last seminar. Definitely his, I should say HPL seminar. But he's leaving us at the end of the year. He's not going very far, and we'll talk about that next. But I just wanted to say a few things because he's been around for quite a while. It sort of feels like I'm losing an appendage here. So Ifaz joined the HPL as an ISI postdoctoral fellow in 2017 after getting his PhD in aerospace engineering from Carleton University. And since that time, he really took on an active leadership role in my lab and just throughout the HPL in general. So just within my group in the last 5 years, he's helped me supervise 11 graduate students and more than 25 undergraduate students. He's really been instrumental in helping with the organization and data reduction on some really big projects, included 2 clinical trials. He's published 10 peer-reviewed manuscripts as first authors based on his research in the lab, and he's co-authored another 7 manuscripts with various graduate students in the lab because of the strong contributions that he makes to all of our research. Within the HPL, he kind of maintains a bit of an informal appointment of the resident expert for computational modeling and imaging analysis, but he truly enjoys helping others solve difficult problems and succeeding. Ifaz was the organizer and chair of the HPL biomechanics seminar for 2019-2020. I think we had to pivot this whole thing because of COVID, but it was he made it a seamless transition. He's also contributed a lot to our faculty. He's taught 263 a couple semesters. He is very happy to give guest lectures in Walter and I's 664 class and Kathy's sports medicine class. And the feedback from the undergraduate and graduate students has always been overwhelmingly positive. They appreciate his pace of instruction, his friendly demeanor, and his positive attitude. So maybe if everyone could just please join me in thanking Ifaz for everything he's done. Let's get to his talk, okay? So when Ifaz was asked what scientific accomplishment are you most proud of, he said, I'm really proud of this work that I'll be talking about today. It was a very large project with a lot of coordination, people, equipment, general timing. So it was a fun challenge getting to do that. So he'll talk about how much of an effort it was and how many people were involved. What are your future plans? What are you working towards? If I could be a postdoc forever, I would do just that because it's possibly the best job in the world. And in terms of what we get to do day to day, that's not realistic though. I'm really looking forward to working with Pershing Snyder's group as a research associate next year. I'm sure he's also looking forward to making more money. And then this is funny. What are some interesting facts about you that are not reflected in any of the above questions? This includes extracurricular activities, hobbies, and personal interests. He says, so after giving 4 other seminars, I have officially run out of fun facts. So I guess without further ado, you can begin your talk, right? Yeah. Titled, Influence of Antiresorptive Drugs on Bone Strength, Fatigue Life, Density in Microarchitecture, Implications for Typical and Atypical Fractures. Thank you.
Thanks a lot for that awesome introduction, Brent. Yeah, so I guess this is sort of, I consider it my magnum opus in a way. Like, it's been many years worth of work, and I'm really, really happy to be invited here to talk to you all about it. So let's jump right in. Everything I'm going to talk to you about today is sort of broadly under the umbrella of osteoporosis research. So as you might be aware, it's a metabolic disease that influences bone across multiple scales. It results in a tissue microarchitecture that's more porous, resulting in a material that's less dense, and a structure that's ultimately a lot more fragile and prone to fracture. And these fractures are a problem. The last data that I was able to see in terms of costs to Canadians puts the price tag at about $4.3 billion a year. To put that into context, that's more than a lot of other diseases that we think of as very important. Things like coronary heart disease or stroke. But it's not just dollars and cents, of course. This is a disease that affects people, and it affects a whole lot of people. The stats that I most commonly see is that it's suggested that about 1 in 3 women, 1 in 5 men are going to experience an osteoporotic fracture at some point in their life. And then when you think about it, depending on where those fractures are, they can be quite serious. So for example, a person with a hip fracture has about a 30% shot of not making it another 2 years after that event. So hopefully I've kind of convinced you, as I am myself convinced, that this is a problem that's really kind of worth our attention as researchers. But of course, as researchers, we're very much interested in understanding the how and why of how things work. And so to really get into that in terms of osteoporosis, I need to talk a little bit about the bone remodeling cycle. As you probably remember, this is a cycle that occurs, regularly in our bodies. It's how our bones access our mineral reservoirs and maintain that tissue. It's also really complicated and definitely still something that's an area of current research. So for the purpose of today's talk, I just want to, on a very high level, draw your attention to what I think are some of the key actors here. So you have these cells called osteoclasts, whose job it is to remove bone. And then they have an opposite counterpart called the osteoblast, which is a cell whose job it is to add bone. Now, these two things, they don't work independently of each other. There's quite a degree of crosstalk where the activity of one of the cells sort of releases signaling compounds that upregulate the activity of their opposite. And in this way, this crosstalk maintains a state of sort of quasi-equilibrium, in that in a healthy person, a young healthy person anyway, you don't necessarily get a situation where there's runaway bone growth or bone loss. You can get subtle differences, and that's how things like functional adaptation happen. But in general, the two forces are relatively close to each other. That's not the case in someone who has osteoporosis. And maybe one way to think about this disease is it causes a degree of decoupling between the processes, and it makes it possible for bone resorption to greatly outpace bone formation. And once we understand that, a possible solution to osteoporosis sort of becomes self-evident. Maybe we can give people drugs to stop or slow down bone resorption. And in fact, that is exactly what we do very often in clinical contexts. There's a whole array of drugs broadly called antiresorptives, whose job it is to do exactly that. In today's talk, I'm going to focus on 2 specific antiresorptives. The first is the bisphosphonate alendronate, which is probably one of the most commonly used ones today. And it works by binding to hydroxyapatite mineral in the bone and basically making it harder for those cells to resorb that tissue. The other one that's going to play a big role today is the rank ligand inhibitor denosumab, which effectively works by interfering with the signaling compounds that are responsible for osteoclast lifespan and formation. Regardless of which of these 2 drugs I'm talking about, in general, the research is relatively clear. These drugs are very effective at slowing down or even reversing bone loss, resulting in stronger bones. And more importantly, those stronger bones result in less clinically observed fractures overall. And that's kind of what I'm showing on this graph here. This was a study that looked at sort of a subset of individuals who are deemed to be at high risk of osteoporotic fractures. And they found that when they gave some people alendronate, the group that got the drug had a relative risk reduction of 51% compared to the people who didn't. Now, this study is not an outlier. I didn't cherry pick it. There's lots and lots of studies on both alendronate and denosumab that'll show you something similar. And because of that, it's maybe not surprising that if we look at especially historical data, these drugs have seen a nice, at least there was a time when these drugs had seen a nice uptick in prevalent use. And this is data from the American healthcare system, so we can generally trust that it's relatively good data. But what I find really fascinating about this is that uptick in antiresorptive use has been associated with a nice downward trend in the number of actual fractures, osteoporotic fractures, that have been observed in clinical cases. So if this was the end of the story, I'd probably be done here. But this is the part where I'm supposed to be describing a problem, right? So here's that problem. Recently, I'd say maybe in the last decade or two, we've seen a downward trend in the use of antiresorptive drugs and a plateauing in the number of cases of osteoporotic fracture. And people believe that if this trend continues, we might actually, for the first time in a long time, see a reversal where people are actually, the number of fractures is increasing. Of course, it's hard to say for certain why this is happening, but it's very strongly believed that all of this is because of recent fears over rare but very serious side effects that can occur, especially if people take these drugs for a long time. And the big side effect that's usually on everyone's mind is this phenomenon called the atypical femoral fracture. So this is a very unique, I'd say, kind of injury that is associated, it's very clearly associated with prolonged use of antiresorptive drugs. What makes it atypical is that it happens on what we normally think of as being the strongest part of the bone. So typically the midshaft or the subtrochanteric region, and these fractures present as sort of transverse fractures of the lateral cortex. Notably, they also happen in situations where there's no or minimal trauma, and the patient may have prodromal pain, or they might even develop something that looks like a fracture callus, so a little bump that shows up on radiographs long before the actual fracture has happened. And these last few points together make people think that these are probably a sort of mechanical fatigue failure. And that idea has led to this predominant or prevalent hypothesis about why these atypical fractures occur. Some people believe that they happen because if you take the drugs for a very long time, it causes detrimental changes that impair bone toughness. And I think that's a really clever hypothesis in that it seeks to explain why taking these drugs increases the risk of atypical fractures, but not necessarily typical ones. But unfortunately, being clever doesn't make it real. And I think it's really important that we keep in mind that that hypothesis is not well supported in the literature. And because of that, we felt it was very important to have a more comprehensive understanding of how these drugs really impact the bone. And that's ultimately what I wanted to talk about today. So I wanted to talk about a whole mess of experiments and measurements that our group did to try and better understand changes in bone mechanics associated with these drugs. Now, bone mechanics is very wide. There's a lot of specific things I could mean when I say that. But with that said, our time and the amount of tissue we have is limited, so we really wanted to make sure that we focused on things that were most clinically relevant. So let me kind of walk you through what that is. Imagine we had some bone. It could be a little piece of bone that's machined out, or it could be a whole bone like this femur here that I've got on the picture. If we wanted to know something about its mechanics, we can put it in a machine and basically push on it till it breaks. If we do that, we might get something that looks like this, a relationship between the force applied and how much this bone has deformed. One very common and very important property is what we call the strength, which is simply the force, the maximum force that's achieved any time during this monotonic loading. And the reason I say strength is really important is because it's fairly well correlated. It's generally known that if you make your bones stronger, your chances of getting an osteoporotic fracture go down. And because of that sort of relatively well understood association, there's been a lot of work to the point where I think bone mechanics has gotten to a stage where we don't necessarily have to put bones in our machine in order to tell if they've gotten stronger or weaker. We know for a fact that things like microarchitectural improvements, improvements to density, or changes in geometry, we have ways of understanding how those things might influence the strength of these bones. But strength isn't the only thing that we can measure. It's not the only thing that can change. I talked about the other property a little bit earlier, and it's called toughness, or sometimes you might hear it referred to as work to failure. And we can measure that. It's sort of the area underneath our force displacement curve. Now, the prevalent hypothesis for atypical fractures says that these bones become more brittle, as in they're not able to deform as much before complete rupture, so the amount of energy they can absorb goes down. And that's a really cool hypothesis, but we didn't do necessarily a lot of work to failure in this work because I don't think a monotonic test is really what we need to be looking at if we're talking about atypical fractures. they're mechanical fatigue failures. So we should be doing mechanical fatigue experiments. And it kind of an example, I'll bring up this photo again later, but you can do that again at either the material or the whole bone level. But the reason that I say that it's really important to do mechanical fatigue experiments is that although we sort of generally understand that if you make a bone stronger and tougher, you're going to probably improve its fatigue life, that direct relationship is difficult to calculate. And if you have a situation where, say, strength is going up and toughness is going down, I don't know any way of trying to figure out what that actually does to fatigue life. So that's why we think actual experiments are really the key here. So with all of those kind of preliminary ideas in mind, let me jump into talk about what I really wanted today. And so my talk is separated into two parts. This first part is going to be all about some of the non-destructive measures that we did in order to understand the strength, density, and the stiffness of a monkey humeri specifically. So why do we care so much about humeral strength? Is there any sort of clinical reason for looking at this bone in particular? I think so. And it's because that although antiresorptives are sometimes considered to be a systemic drug in that they are delivered by your circulatory system to all the different bones in your body, we do know that the actual magnitude of the effect and the benefit you get really does seem to vary site to site. In general, it seems that like very central sites like your spine are very easy to affect, and you get less effects in more peripheral sites, especially the upper limb. And that's kind of a problem because humeral fractures are relatively common, and they are associated with high rates of morbidity and mortality, so they matter. But they're also a site that's not very well explored in the literature. So for example, we know that a drug like denosumab reduces the rates of humeral fracture in postmenopausal women. We don't actually have any supporting clinical or preclinical data of what the drug does to bone density at those sites. So something like that is a gap that we were really well positioned to fill out. So that brings me to exactly what this specific study is. The purpose of this study was to look at the effects of denosumab, alendronate, or combination thereof on the humeral bone of cynomolgus monkeys. And then we used both high resolution computed tomography and regular clinical resolution CT to understand the changes in the bone. And then we combined that with finite element modeling to understand how those radiographic changes influence bone mechanics. We were, I think, very lucky to be able to use a monkey model for this work because the monkeys have a lot of really key similarities to humans that make them a very powerful model for osteoporosis. A few of the key ones, for example, are the fact that this is an animal that undergoes intercortical bone remodeling. So it winds up with microarchitecture that largely resembles a person's. This animal in captivity is also known to experience menopause and the bone loss that comes with that. And because of that, ovariectomy of these animals is a very well accepted model for osteoporosis. And typically, these animals, I do acknowledge that these animals are typically very expensive to keep and sometimes a little bit dangerous. And because of that, we often reserve these monkey models for studies that are really on their final step before clinical trials. With that said, I definitely can say we're not ramping up to a clinical trial necessarily of any kind. We were able to use a monkey model because this was a secondary use protocol. We got to use some tissues that were banked from a previous study. And specifically in that previous study, they had 45 animals that were treated for 12 months with either alendronate, denosumab, or some combination thereof. And then in the work I'm going to talk about, we used humeri that were harvested and we used those for our analysis. Before I show you a little bit more about specifically what we did, I do want to give a quick shout out to Elena Stahl, and I think she's in the audience here. She did some really fun supporting measurements for this work. Basically, the contralateral limb to the ones that I analyzed had previously been, the animals were treated with a marker of bone formation, and those bones were fixed in formalin. So Elena was able to go in and process that tissue and both visualize and quantify basically how much bone formation was going on when we were treating these animals. Now remember, formation is coupled to bone resorption, so we think that this is really a good indicator of just how much turnover or remodeling was really happening in all of these groups. The thing I want to draw your attention to, and Elena definitely had really nice quantitative figures, but I just wanted to show you some pretty pictures here today, In general, what I want to draw your attention to is this idea that the vehicle groups, in the vehicle groups, we saw a lot of remodeling. In the alendronate group, we saw much less, but still some remodeling. And denosumab, it seemed, pretty much killed the remodeling altogether. So this was really exciting for us because it suggested that the drug was doing what it does in people and what we think it should be doing in these animals, biologically speaking. So hopefully we would see that reflected in our measurements. And now we can finally talk about the first set of measurements that we did. So we started off, one of the early things we did was we took some high resolution micro CT images of the proximal part of the humerus, a part that we weren't going to test later on. We performed some image analysis where we separated the cortical and trabecular regions, and we looked at tissue mineral density, bone mineral density separately in these regions. A big shout out to Ying Zhu from Steve Boyd's lab, who really sort of developed the protocol and was largely responsible for the analysis, although I definitely helped out with the legwork, so I am confident in what we did. After we did that analysis, we also went on and we did some finite element simulations. So I was really wondering how much to talk about for this part because the grad students in our group know that I can talk for hours about finite element simulations. I'm definitely not going to do that here today. Maybe just a high-level talk is more appropriate. So effectively, this is a technique that lets us understand mechanically what those radiographic changes are doing. And the way this simulation works is that I take geometry information from the scans, and then I apply sort of assumed reasonable material properties for the bone tissue. I then tell it how I think this section of bone might be loaded. So for this particular analysis, I held one end and we kind of pushed down on the other, a fixed amount of displacement. And then we used the principles of continuum mechanics in order to sort of understand how the structure, how the bone would locally and globally react to that sort of loading environment. In this particular work, I then took that information and I got a measure to estimate overall stiffness and strength of the bone. I think our results for this part especially were really clear, and they really did match our expectations. So the first thing we saw is this plot here, which is a plot of cortical porosity across the different groups. And we saw clearly that treatment with drug, and particularly the vehicle DMAB and the DMAB alone groups, had much lower porosity than the animals that only got vehicle. We also saw some nice improvements in tissue mineral density. And in this case, it was the DMAB alone and the 6-month alendronate and 6-month DMAB groups that had sort of the statistically significant post-hoc comparisons. And given that bone mineral density is just a function of the tissue density and the porosity, we really weren't surprised, although it was a nice little sanity check to see that overall bone density was higher in all 3 of these groups that got identified. The finite element models told a pretty consistent story as well. In general, we saw increases in finite element predicted strength, which is the plot here on the left, as well as stiffness. And the intergroup comparisons were really predominantly significant for the DMAB alone group. So at this point, we had some idea now of what this drug was doing. But of course, that analysis is limited to just one very small part of the bone. We wanted to get a picture of what it's doing across this whole structure. And perhaps a more appropriate technique for that is clinical resolution CT. So in this work, our typical workflow was that we identified the bone using a sort of semi-automatic technique based on thresholding. And then all of our scans had an object that looks kind of like this. It's a calibration phantom. It's basically an object that has 3 little rods of known mineral density, and we're able to relate the CT intensity, so the color or the brightness of these rods, to an actual density value to assign to our bone. After that, we divided our bone into 4 different regions: the ultraproximal, proximal, 1/5 proximal, and midshaft, and then further subdivided those into cortical, trabecular, or integral compartments. Just like before, our key measurements here where we were interested in trying to figure out bone mineral density and bone mineral content. In addition, again, we did some finite element modeling, although the process for this one's a little bit different than before. To make these models, we took our scans and then we downsampled them to an isotropic voxel. And then we turned basically all of those individual voxels into little cubes that form our finite element mesh. When you're doing something like clinical resolution CT, we can't see the individual pores as well as we used to be able to do. So in order to compensate for that, what we do in something like this is that we assign heterogeneous material properties based on the density variation, which is something that we can measure. In addition, there's some nice complexity in these models that I'll just gloss over very briefly. Our models were anisotropic, just like real bone, and we also had nonlinear material behavior. So that is to say, we were sort of modeling what happens to bone as you approach yield, the fact that stress just doesn't increase infinitely as you keep pushing on it. And in this case, we used, again, we had to put some loads and boundary conditions on these bones. And since we wanted to get a sense of what's really happening here, we decided to put an array of loads and boundary conditions. So I did simple torsion, compression, and bending, which we thought was a nice representative sample of all of the different ways that a bone can be challenged. Our results here were quite interesting in the sense that, well, just like before, we definitely did get to see, or we definitely did detect differences in cortical density. And that's kind of what this graph here is. This one happens to be at the midshaft of the bone, which is where those differences, I think, were easiest to detect. But we generally saw differences across all of the regions of the bone, and that's kind of what I'm showing in this table here. You don't necessarily have to worry about kind of focusing on the individual numbers so much, but I do want to draw your attention to a few of the trends. In general, it seemed like the differences were largest and easiest to detect near the midshaft and a little bit harder as we went up the bone. And across the way, although my intergroup comparisons didn't come out statistically significant, in general, it seems that DMAB tended to have bigger differences than vehicle, which meant that there were often cases where we could detect that difference in the DMAB group when we couldn't in some of the other groups, just like some of the data I'd been showing you before. With that said, this is just a very small number of measurements. We did a lot more. And I don't want to show you the full table, but I will draw attention to the fact that we didn't see differences in trabecular regions, in geometry, things like cross-sectional area, or in bone mineral content. And I think it's that last one probably explains why we didn't really detect any differences in our finite element measures here. So I know that was a whole lot of measurements, so let me just really give you kind of a brief high-level summary of what I talked about here. In general, we were able to detect differences in especially the cortical bone of animals that were treated with antiresorptive drugs. Micro-CT showed lower porosity and higher density, and we also saw higher density in our clinical resolution CT. In general, the intergroup comparisons weren't significant, so I can't necessarily say which specific antiresorptive treatment, or I can't statistically say which specific antiresorptive treatment was best. But I do note there was this really obvious pattern in that the 12-month DMAB group tended to more often have the bigger difference versus vehicle. And it was definitely the one that was more often pinged or detected as a difference versus vehicle. And I think that's really interesting because all in all, it means that if these similar patterns are mimicked in humans, especially in this idea of improving bone mineral density of the humerus, it probably explains why use of denosumab is associated with reduced risk of humeral fractures in women. However, I will end on this note is that we were certainly surprised that we didn't see these trends in bone mineral density reflected in things like bone mineral content or the clinical CT measures of finite element models. I don't think my conclusion here is that antiresorptive treatment doesn't make bone stronger. I think really what's happened is that the design of our study, the cross-sectional nature, wasn't able to separate the between subject differences in size and shape from the treatment effect. So that's sort of my kind of personal opinion about that. So with all that said, let's jump into what I think is a really exciting part, which is our fatigue analyses in part 2. The general purpose of this part of the study was to use a overreactomized, again, it's the same Sino-Milgus monkey model that we already used, but this time we were looking at bone fatigue life differences. And so we did that by harvesting small samples, matchstick-sized pieces of bone, and loading it cyclically until failure in a bending configuration. We also did some more supporting measurements to try and explain or understand why we were seeing what we were seeing. And so we did, again, some more high resolution micro CT, as well as some traditional, what I'm going to call manual density measurements to really look at what's going on. And I want to say at this point in the work, because we'd already seen everything from the previous part, I don't know if I speak for all of my co-authors, but I'd say that I kind of had this hypothesis at this point that I didn't think that we were going to see less fatigue life in antiresorptive treated animals. I think all of those changes that we saw earlier are going to result in more fatigue life in those animals, but we'll see if I was right or not. Just a recap. Again, it's the same animals as before, so the same 5 treatment groups, although I will draw attention to the fact that the numbers have changed a little. In this part of the study, we used a subset of animals so that we can balance all our different loading conditions. Before we were able to test these bones, though, we had to quickly cut up some samples. And when I say we, I mean Andrew Sawatsky, who did all of it. And when I say quickly, I mean that this is very time-consuming, tedious, and challenging work. And I think it took him like a year and a half, 2 years or something, Andrew? Yeah. But the reason it took so long is that we were very interested in making sure that things are very uniform so that we had a good test here. We got samples evenly from both the midshaft and the 1/3 proximal region, and we took them from all around the 4 corners of the bone. All in all, across all of our different animals, we had 240 samples that were eventually tested in this study. And so let's talk a little bit about those tests. I kind of showed you the picture of it before. We did a simple 4-point loading configuration, and we tested them until either rupture or a predefined runout of 1 million cycles. All of those samples that we pulled out were evenly divided based on both the harvest site, the animal, and the group that they're from across 4 different load levels. So 80, 100, 120, and 140 megapascals of sort of peak load at the cyclic peak. Before I move on, I want to kind of draw your attention to something. If a sample survives 1 million cycles, that's about 6 days of testing. So with 240 samples, if they all ran out, that would have been something like, I think, 4.5 or 5 years of nonstop running our machine. Thankfully, they didn't do that, and I only had to run the machine nonstop for about 2.5 years. So in addition to recording the number of cycles to failure, we were also able to, of course, track the stress and strain across the cycles. And from that, we got some biomechanical measurements like stiffness and the total energy dissipated as part of this failure process. In addition to that, as I mentioned earlier, we did some micro CT. So for this study, I developed some custom code that let us sort of automatically identify the pore space. And then I got to use an algorithm that was developed, or a workflow that was developed by Lindsay Lowndegan for her PhD work that quantified the porosity and things like the pore diameter and the pore spacing. We also, since we had little pieces of bone, we were also able to take what I'm going to call some manual density measurements, just with calipers and scales, and we even had a little oven, so that meant that we were able to separately quantify the apparent, the dry, and the ash density of these bones. We also looked at something called ash fraction, which we think is really indicative of composition of these tissues. So let me show you the results from all of that. And I think this was really cool and maybe surprising, depending on whether you believe my hypothesis or not. But in general, we did find that antiresorptive treated animals survived longer than the ones that did not. So I know there's a lot going on here, so let me walk you through what this particular graph shows. The different clusters are the different load levels. Each color is one of the different groups. Notably, red is vehicle and black is denosumab for 12 months. And then you can kind of see that there appears to be a nice trend here. That trend, I should say before I move on the statistics. Statistically, we saw it was 12 months of alendronate followed by DMAB, or DMAB alone that had statistically higher fatigue life. But that trend, I think, is a little bit clearer if we plot the data in something that looks like this. So this is just the same data. It's just plotted on a log-log scale with stress on the y-axis. And the solid lines here are the result of a survival analysis fit of the data. So special thanks to Andrew Pohl, who's here as well. He developed this algorithm for some work with Lindsay Lowndegan in her PhD as well. But you can kind of see the trend here, I think, really nicely, where the red line indicating the vehicle group is on the left, which means the lowest fatigue life of all of these. And then the other treatment groups, especially denosumab alone, is kind of really far on the right, indicating that it tended to survive the most. Follow-up measures all sort of supported those fatigue life findings that we saw. In general, we saw higher initial modulus in the denosumab group, as well as, surprisingly maybe, more energy dissipated, not less, in the denosumab-treated animals. We also saw our micro-CT also sort of was really consistent. It's nice because you don't always get measurements that are so consistent with each other, but we saw that all of the antiresorptive treated animals had lower porosity and a greater, basically smaller pore size of it, or smaller size of individual pores compared to a vehicle. Our density measurements were also pretty much consistent with things in that we were able to detect increased density in the antiresorptive treated groups, particularly the denosumab alone group. And I think it's really cool that we didn't see a difference in ash fraction, because I think that would seem to indicate that what these drugs do is they don't necessarily change the composition of the tissue, not the ratio of organics to mineral, but really their effect on density, I think it seems, is more based on what they do to the pore space of these bones. So we're near the end of the talk, thankfully, or maybe thankfully for you guys. I've had a lot of fun up here. But again, let me just end with a little summary of everything that I talked about, because I know I just bombarded you with years worth of measurements. So contrary to the prevailing hypothesis, we did not see any detrimental effects after 12 months of antiresorptive treatment. And in fact, we kind of saw the opposite. So we saw that antiresorptive treatment and that 12-month denosumab group, again, in particular, survived more cycles than the vehicle-treated animals. And those differences all seem to be really well explained by what the drug was doing to porosity, pore size, and density. Clinically, I think this alone is really interesting because people are starting to think that there's a subset of clinical fracture cases that really have a mechanical fatigue component to them. So we think that these data like these might explain why antiresorptive drugs are generally have been in the past considered so good at reducing the rate of clinical fractures. With that said, I think the key here is that we did not observe any of the detrimental changes, or really anything that could directly explain the association between antiresorptive treatment and atypical femoral fractures. In fact, I want to kind of remind you that that's really interesting because in terms of the biological effect, it's doing what we think it should be doing to the bone. It's doing what we know happens to humans. So how come we don't see an association in these animals, and you do see it in clinical cases between prolonged treatment and atypical fractures? I don't have an answer for that, and I think that's going to be a fun thing to research for the rest of my life. But I do have some ideas, and maybe I wanted to discuss those with you for now. One idea is that it might be that the association between atypical fractures and drug use is really just correlative. And so a few weeks ago, Brent did a talk where he introduced really nicely this idea of hip survival bias. And this hypothesis basically says that the drugs are so effective at reducing osteoporotic fractures that suddenly you have this subset of people who normally would break their bone and just not be able to ambulate anymore. And now they can continue loading their bone, and eventually they wind up getting a fatigue-like failure somewhere down the line. I think that's a really cool hypothesis, but I also want to accept the fact that it's entirely possible there is a mechanistic relationship that's somewhat outside of the scope of what we were able to capture in these tests. One of the things that I think opponents of this idea are really going to fixate on is the fact that we couldn't really test animals for more than 1 year. It's very, very challenging, of course. Many of you probably know in the audience, to have animals, it's very expensive to have them for that long. And so it's certainly possible that if an organism gets this drug for much, much longer, that you start to see detrimental effects pile up and sort of overcome the benefits of the drug. Certainly possible. I can't say that's not happening in real life, but one of the things that bugs me about that hypothesis, and it's always kind of bugged me a little, is that if strength is staying the same, but toughness is going down over time, Well, things like falling also depend on toughness. So I'm really surprised that we wouldn't necessarily see, and we don't see an increase in the rate of typical fractures alongside atypical fractures in people who take this drug for a long time. So that makes me kind of hesitant to say that this is really what's going on in reality. Where I probably put a little bit, or what I think is maybe a little bit more likely, is the notion that atypical fractures in humans are very, very rare. Our model that we've been talking about here, it's not a model of atypical fractures specifically. A model like that doesn't exist. We don't know enough about these fractures to reliably cause them in a laboratory environment. This is a model of just the average response of an average, or we think it's a good model of the average response of an average person to antiresorptive drug. And it's entirely possible that given how rare this condition is in people, that there's something unusual about those people who get it. Maybe their bones are a little different. Maybe it's mechanical, maybe their bones are a little different. Maybe they walk funny, or maybe it has nothing to do with mechanics. Maybe their individual response to the drug is something that's a little bit different from what we consider the normal or the average person that we can capture here. I don't know the answer to that. It's a really hard question to solve. And if anyone has ideas on how we can test that, I'm definitely open to it. With that said, that's everything. Thank you so much. That's everything I wanted to talk about. A brief disclosure statement. This work was part of a investigator-initiated grant sponsored by Amgen. They were the ones that also sponsored the original study, and they were the ones who gave us the tissues that we analyzed. Big, big thank you to my co-authors and our lab group, and to everyone here for listening to me as a captive audience for the last 45 minutes or so. And I'm happy to answer any questions. Thank you so much.
Start us off. All right. Thoughts. I know you've done some previous work on looking at what characteristics differentiate people with AFF and those who don't, like the bone stuff. Could you maybe speak to what other characteristics might predispose people?
So the thing that's really well known is that, or the thing that we think we know really well, is that it seems that people who get atypical fractures have much more curved bones. So we call that bowing. So basically, instead of being a nice straight bone like this, it kind of bends like that. We know that seems to be something that we've observed clinically. Early on in my time here, I did some modeling to demonstrate what you kind of intuitively would suspect is that when you have a very curved bone, that causes higher strains in kind of the middle of the shaft, the area where these fractures are often occurring. I think that's all we can really say conclusively that is different about people that get these fractures. With that said, I do want to stress a lot of people with straight bones also do get, or you can still have a perfectly straight bone and get atypical fracture. I've definitely seen cases that look like that as well.
Hi, Ifaz. Thanks for that presentation. I have a question about the variability in response to drug treatment. So you don't have pre-post measures, obviously, you're just taking these samples at one point. In humans, we'd expect a huge variability in terms of bone mineral density. So just seeing the BMD measures wouldn't necessarily be indicative of the response to drug treatment. So in these monkeys, is BMD very homogeneous? Is that spread in the data an artifact of having different baseline BMD, or is it an artifact of just some not responding well to drug treatment?
Yeah, so I can say that in the parent study, that was longitudinal. So they were able to separate out those sort of between subject differences in baseline density versus what the drug is doing. And other studies, not necessarily just in cyno monkeys, has looked at that effect as well. So I think it's really, I can confidently say that antiresorptive drugs do in fact increase their bone. What we're seeing is not purely an artifact of just that we had some animals that were bigger or stronger or anything like that. In fact, the parent study also did some baseline measurements. And as far as I can remember in terms of weight, initial modulus, or so they didn't have initial modulus, but in terms of starting characteristics, nothing that jumped out as a red flag that would suggest, oh, this is goofing up our ability to see something in the study. So that's kind of where I think if anything, I think that all that variability that we had makes it harder for us to find statistically significant conclusions. And I think, for example, that's exactly what happened with the clinical resolution CT images.
Questions? All right. I'll go next.
Okay. Can you compare how alendronate denosumab, like what their action is on bone, or something about how it's delivered that would cause denosumab to have a greater effect?
Oh, okay. So in terms of their action, it's that alendronate, it basically makes the bone tissue harder for the cells to resorb. It doesn't stop it necessarily completely. Like they can still resorb that tissue. Denosumab stops cells from being born. So at certain doses, like the doses that we're doing here, which is actually much higher than maybe you might give a real person, you can pretty much just stop remodeling altogether. In general, and again, this is very dose dependent, right? Because you can manipulate dose and get, I think, anything you wanted. But at the doses that are given in clinical contexts, I've seen sort of head-to-head comparisons that suggest denosumab is the more effective one at slowing, at maintaining bone density or improving it versus alendronate. But again, that just depends. If you up the dose, you could probably see the opposite if you really wanted to. Did that answer your question?
Yeah.
Kind of related. Do you think that like an injectable biphosphonate would change anything if it's more like localized?
Oh, these ones were all IV
Oh, okay.
or sub-Q. So these ones happened to all be injected. In people, I can't really speak to in terms of like drug dynamics, if that helps. I know that injectable bisphosphonates like zolendronate are nice because taking something like alendronate daily is a real pain in the butt because you have to, like it causes lots of gastrointestinal side effects. So it's a real pain to do that every day. You can get better compliance if you give someone a shot once a year. In that sense, I think there's a benefit. I don't know if there's also a benefit in terms of how the drug gets to your body.
Did I understand correctly that these atypical fractures, they occur at a specific place, usually?
Actually, surprisingly, I would have said yes, but having looked at a lot of data, I'd say no. And the clinical definition now is that it's basically anywhere below the lesser trochanter and above the flare for the condyles in the bottom can be an atypical fracture. And I've certainly seen them happening all throughout, although I'd say the top half is more common. A fracture really down near the condyles is much more rare. I think I've only seen one in the hundreds of pictures of them I've looked at so far.
So could it be that when I take this drug and then I have a greater bone mineral density and my bone is harder, that structurally the bone would adapt because it would like to see a certain amount of strain all the time? And therefore maybe the strain would be similar. And you did your fatigue testing with applying a certain amount of stress. Do you think you will get the same result if you apply a certain amount of strain?
Okay, so the first part is, I'll answer that in parts. The first part is, do I think that eventually you get adaptations that result in you have a harder tissue but just less of it? As far as I know, these drugs aren't associated with that when you correct for things like, when you correct for age, of course, right? Because the person who's had it for a long time is just naturally aging. So it doesn't seem to, because, and that makes sense, right? Because that process by which the bone would adapt, that is the remodeling process that's being interrupted by the drug. So it effectively stops that remodeling that would normally be happening. So I think that's why you don't see that. In terms of our specific experiment, I thought it was really good that we did stress rather than strain, in the sense that by doing stress, we're saying that this, and I think that's really representative of how people move, and that you have a certain action or a certain activity, regardless of how strong your bones are and how much they can respond to it. It's the forces that you prescribe based on what you do, and it's your body's job to accept the strain that comes from that. So that's why I think a stress-based criteria was better. If I do a strain-based, I actually did calculate it. I didn't show it to you. But if you look at just fatigue life as a function of tissue strain, you suddenly don't see any effect of drug treatment at all. All my data lines up on top of each other. And I think that makes perfect sense. It says that what this drug is doing is it makes the bone better able to accept the forces that you're putting onto it. It's not fundamentally changing it compositionally in a way that the strain relationship between strain and fatigue life is different.
That's what I would have assumed, that the bone, when you give it a drug or you starve or you eat a lot of protein or whatever, you change essentially what you give to the bone and it can become stronger or stiffer, so that maybe it would adapt because it likes to see a certain amount of strain. So if I run a lot and put a lot of load on it, that my bone will get stronger, so that the everyday strain you would see would be similar to a person who walks every day.
Exactly.
And so then that could be potentially an argument for doing a strain test rather, a strain control test.
Yeah. So in the case of a person like that, basically, what's happening is you are choosing to put more stress on that person's bone or on your bone, and that's why your body is compensating in a way such that the strain it sees is lower. I think because I think the general notion of whether changing strain influences things, we can already get out of this data, because that's the nice thing about having a wide range of bone densities and stresses, is that we can absolutely look at it in terms of strain. I think the open question then is whether force differences between people can explain atypical fractures. And actually, I don't know, but I know that one of the clinicians that we work with, Angela Chung, thinks that people who have atypical fractures might have different muscle strength or walking patterns or things like that, but we haven't been able to really test that theory yet. But I think that's where I would look. I wouldn't necessarily, my next test of that wouldn't necessarily be more material stuff. I'd want to know what's happening in people.
Do you know if the drug injection potentially altered the behavior of the animals? Were they more active, were they less active? Was there any observational data on that?
Oh, I don't know, because I didn't get any observational data on that. I know that they didn't have a non-injected, a completely like a non-touched group of animals to be able to draw those conclusions. So it would really, if it happened, it would have been noticed maybe by a person handling it, but it definitely wasn't recorded, and I haven't seen that.
And your control animals, they were also overrectomized?
Yes.
And did you also have a non-overrectomized control there?
No, for this one we did not. Because, and I think the justification for that is that these drugs really aren't used on people unless they have severe osteoporosis. Actually, one of the things that Anna in our group is interested in looking at is how do these drugs behave in the absence of overectomy, and maybe she'll be able to talk about that in a few months. But
yeah, no, I was just wondering, how much osteoporosis do you actually induce through the overectomy?
Qualitatively, a whole lot. Quantitatively, that was definitely one of the things that they quantified over the course of the parent study. So they did look at BMD and things like that. I don't remember the magnitude of the numbers, but clear, and it's big. Yeah, it's big.
I got a question from Robert. So go ahead, Robert.
Ifaz, that the autonomic nervous system has a role in the control of bone remodeling directly. And I very, very rarely hear any input. I hear about the drugs. I'm wondering if these drugs have a direct action on the osteoclast and osteoblast, or whether in fact their action is via influencing the sympathetic nervous system. And if not, why do we not hear more about methods of influencing the sympathetic nervous system in order to reduce osteoporosis? We just hear about the drug side, unless the drug side is doing that. Are you familiar with that?
Yeah. Thanks, Rob. So I'll say in terms of what the drugs do, Denosumab, I'm very confident that people are very keenly aware of the fact that, yeah, its job, or its chief job, or one of its major things in the body is it is in fact the compound that's responsible for osteoclast genesis. So when you inhibit it, you simply make less osteoclasts, and I think that pathway is relatively, I'd say, relatively well understood. Whether any of these drugs have like, they also affect the nervous system, I don't know. I haven't seen that hypothesized, or I haven't looked for it specifically, but I haven't seen any mention of that in discussions of stuff that I've seen. So I'm gonna say maybe not. As for why people don't design drugs specifically to do that, my understanding of designing drugs is we don't necessarily know enough yet to think of an effect and then think of a compound that might cause it. We kind of go the other way around. We know certain compounds have certain effects, and then we kind of go from there. So it might just be that we haven't accidentally stumbled across the compound that inhibits the nervous system without killing you and changes your bones.
Yeah, I'm wondering if in people who get osteoporosis, if they have a decline in sympathetic nervous system activities in some way, and whether you wouldn't see a correlation in other areas. So people who are prone to osteoporosis, if you can see something in heart rate variability, which also mimics changes in the sympathetic nervous system. And I'm wondering whether it's an avenue of low activity type behavior with older age, where your sympathetic nervous system is not very active, whether there are correlates elsewhere other than in bone that might signal this is your whole behavior problem, and it's showing up through the nervous system.
So I'll say I haven't read anything specifically on that topic. I don't know if osteoporosis is associated with reductions in your sympathetic nervous system, outside of the fact that it's associated with the complex process of aging, and so all of these things are happening at the same time. That the degree of that link is not something I've really looked in. I haven't read about, and I don't see a lot of people doing research on it. And then I haven't also looked for it specifically, so it hasn't come up yet. So, but that might be something to think about in the future.
I'll send you a couple of reviews that do talk about osteoporosis and autonomic nervous system.
Cool, thank you.
So you,
I was just wondering if there's a difference between the clinical outcomes of patients with the atypical fractures and those typical fractures?
Oh, I can't say head to head. I know that Angela's exact quotes on her ethical things is atypical fractures are dramatic and life-changing. So they're really bad. I'd probably put, I don't know head to head, but probably around the ballpark of how bad a hip fracture is, I would assume.
Right. So then, like, can you explain the psyche behind clinicians avoiding prescribing them out of fear of a risk that is the same thing they're trying to avoid by prescribing them in the first place?
Yeah, at the risk of being too controversial, I think it's the same psyche that makes people avoid vaccines. Ultimately, it's a question of, I think we don't like the idea that, and then this is just me guessing, right? Because I don't necessarily, I'm not a clinician, so I can't say what's taught in medical schools or anything like that. But it's my interpretation of it is that we don't like the idea that we're doing an intervention and it might have a negative impact. Even though I think personally, if you were to ask me, I think the risk of a osteoporotic fracture is so much higher than the risk of an atypical fracture. I would rather, I'd take the drug.
just to give you a chance about, we talked about finite element modeling. You made a very nice thing there, wording, you know, that you put in assumed reasonable properties. How do you do that? how do you assume reasonable properties? Like, what's the actual procedure that you use to do that?
Oh, for bone tissue, there's effectively just one, or it's basically just a number from literature that's very commonly used. I don't remember the exact number that I used for this particular study, but I do know that, especially for the type of design of this study, we're interested in differences between groups. And so even if I get that number a little wrong, I suspect I'd still be able to see group differences. The bigger question is whether the material property between animals should have been a little bit different. Potentially, and we tried to get at that, not necessarily through the finite element model, but that's why we did try to measure tissue mineral densities. So if anything, I'd say that if I'm missing something, the effect of the drug should be bigger because my models didn't take into account the fact that denosumab's tissue mineral density was a little bit higher as well.
Did you actually try to finite element model that slab? The slab that you use, the bone, the bone tissue rather than the entire bone. And then you use the mechanical testing on it, and then you would have been able to see exactly whether or not it fit.
I'm not sure what you. Whether we tried to match the some sort of gross material properties. So for the high resolution models that we did, especially that first stage, we didn't mechanically test them afterwards, but that is absolutely something we could do and I know that's been done in the past to get kind of a better idea of the individual, at least average modulus for a person.
And the other thing that I didn't quite understand is, you talked about these atypical fractures in the femur, is that correct? Yes. But then you did testing on the humerus, and you said there is a lot of humeral fractures in people, but I cannot imagine that you have fatigue fractures in the humerus. I can see that you fall and have fractures, but am I missing something there?
Oh, no. So I'd say I like to separate out our data in terms of interpretation, and that I think that first half, the humeral fractures bit, it really tells us a lot of the strength, but I think it tells us something about typical humeral fractures. But the fatigue data, I don't think necessarily is necessarily super relevant for humeral fractures, other than the fact that people think that there is a subset of what might get classified as a fragility fracture or an insufficiency fracture that might have a fatigue component to it. Now, it would have been ideal for the atypical fracture stuff if we had a femur specifically to test. We did not, because those femurs were used up in the first part of the study, so humeri were all that's left. With that said, in terms of mechanically or sort of fundamentally the processes that we want to look into and test, I think there's enough similarities in the sense that the humeri also sees damage, it sees remodeling, it's actually loaded quite a bit in these animals because their cages have to be big enough for them to hang, so that it's not like it's a completely unloaded bone or anything like that. It's as good as we had for atypical fractures.
Monkey, I can see that, but in humans there is no real humeral fatigue fractures. Is that correct?
It's hard to say. There are things that get classified as insufficiency or fragility fracture. And so there was a really nice opinion piece or review paper that says strength is not enough, where I forget who wrote it now, but the opinion of the author was that it might be that a lot of things that get classified are really not, even though there's a final insult that causes the fracture, the reason that you got it in the first place is because you had some damage buildup from fatigue. I think that makes a lot of sense, in the sense that the strength of bones, even very osteoporotic bones, is often higher than what modeling suggests you'd see during something like a fall or a typical activity. So I like that hypothesis, but it's really hard to test. So I'd say, I don't want to say that fatigue doesn't affect the humerus. We're not sure to what extent it does. And I guess typically we don't really focus on fatigue of the humerus, certainly.
I'm wondering if you have any opinion on whether bone remodeling is a good thing late in life, because, well, I think the normal story about bone is that remodeling is a good thing. It's important for development and all that stuff. But then it sounds like, at least for maybe people of a certain sex and a certain age, that you're better off without it. And I'm just sort of putting words in your mouth. It sort of sounds like that. So I just wondered, do you have an opinion on remodeling? Are you better off without it?
Sure. Well, I think in terms of remodeling, I would be very hesitant to stop it completely because it's also so important for homeostasis. It's how, like I sort of alluded to earlier, it's part of how your body accesses your calcium reservoirs from your bones. So I'd be very hesitant to say, let's stop this biological process completely. I think the problem is that remodeling later in life gets wonky or confused is maybe a way to put it, where it's not doing what it's supposed to, which is building bone back up to match what it's taken out. And I think that's where the problem is. So I wouldn't necessarily say that remodeling as a whole is bad. If anything, getting remodeling back to what it was like when you were younger is maybe the right thing to be doing.
Last question, in your raw data traces of the fatigue testing there, it seemed that you had sometimes little steps there. So are those already failures that occur during the fatigue testing, and that it still is able then to absorb more load?
Oh, are you talking about this one right here? Oh, sorry. Is this the one you're talking about?
Yeah.
Okay, so this is actually nothing to do with our specific experiments here. This was just some data I had of mechanical testing for my PhD work. So what I think these little peaks, these little dips are, is I think that's exactly what it is. It sounded to me like I could hear little cracks happening, and I think they're localized failures. And my finite element models suggested that you can get localized failures and stress redistributions that sort of compensate for those. And I think that's exactly what was happening there. So not really related to any of the data I showed you necessarily, but I think you hit the nail on the head. I think that's exactly what those represent. Local failures of the whole structure. And then the rest of the structure sort of compensates for that. You hear it go snap, crackle, pop when you test it. Yeah, you definitely do. Sure.
Oh, so in the density measures, with the different, where you look at the different levels and that was
Could you remind me which one? Because I had sort of 3 different sets of density.
You know, ultra proximal, proximal.
Okay.
So the question is, so if bone remodeling mostly occur, or is dominant in trabecular bone, why do you see the biggest differences?
Oh, that's something that bugged me for a while because, and as far as I'd known in the past, it was that the drug and osteoporosis has bigger effects on trabecular bone because it has more surface area. I think it boils down to our ability to detect differences, in that I suspect there's large differences in volumes and stuff in trabecular regions, and whereas especially the midshaft is a region that's easy to detect, easy to identify, and doesn't change very much across the specimens. I bet that's what it's related to. Yeah, I think so, but I don't really have a strong answer for that. I'm sorry.
Sure, that's okay. And I guess it makes sense when remember when we talked about like how hard it was to segment out the proximal.
Yeah.
Yeah. Any other questions? All right, so let's thank our speaker again.